Bismillah ar-Rahman ar-Rahim. Assalamu alaikum dear all students. Welcome to the next topic of infectious disease diagnostics. Today's topic is about the isolations of different microbes. So in today's lecture we will learn about the different media which are used for the inoculation of microbes, and then the incubation of the media and microbes, and then the reporting of cultures. So the inoculation of different media which are present in the petri dishes. So the technique which is used to inoculate media in a petri dish, it is important that this inoculation must provide a single colonies, because these single colonies are very helpful in identification and to best examine the growth characteristics of any microbe. So single and isolated colonies are important for this examination, and it must also show whether a culture is pure or it is a mixed. So if a culture is a pure which consists of a single type or organisms, so that is called a pure culture. And if the multiple organisms are grown and they are have a different size and types of growth colonies, so these are called the mixed cultures. A pathogen must be isolated in pure culture before it is identified, before it is reported, and before it is tested for antimicrobial sensitivity. Because if we do not isolate a specific microbe, or we do not isolated the pure culture of microbe and perform their antibiotic sensitivity testing, so that antibiotics testing is inappropriate, and our results may have and our report may have a bad impact, or it can even lead to the death, and it can lead to the microbial resistance or abuse of different antibiotics. So that can create another issue. So therefore a pure culture is important for isolation and identification and the antimicrobial sensitivity testing. The inoculation of media in a petri dish is called as a plating out or looping out or streaking out. So these are the terms which are used for the inoculation of the media, and it is not necessary to use the whole plate for every specimen, but depending on the specimen load, a single plate can be used for more than one specimens, and plates can be saved by using half or even third of the plate, especially when there is a specific media and the media is selective one and the microorganisms is very unique. So the area of medium used must be sufficient to give separate colonies. So this is important, that the medium should give us separate colonies, so such amount of area should be used. And before inoculating any plate on of the culture medium, the surface of the medium must be dried, because if the surface of the medium is not dried, then a single colonies will not be formed, or if the surface is wet, then there is a chances of contamination. So our media may be contaminated, or a single colony will not be formed, which can disturb and distract our identification and the colony morphology. Therefore the medium should be dried, and for this purpose First, remove the lid of the culture plate and of the media plate, the petri plate, and then place this petri plate is face upward on an incubator shelf, and the lid is slightly removed. Invert the base which containing the medium and let it rest on an angle on the lid. So the lid is placed downward and the medium, the base containing the medium, it is placed above this lid and in upward direction. So give it 30 to 40 minutes at 35 to 37 degrees centigrade, so that the surface of the agar plates they are dried, or sufficient time so that the surface gets dry and the inoculation and the growth is good for identification. For inoculation, first the loop is sterilized and then allowed to cool before streaking the agar. So before plating, before streaking, before looping out the loop, which is usually an aluminium material, so that is first heated, sterilized and then allowed to cool. It is noted that this should not be cooled with any, do not blow the air or do not place in the path of the air, but allowed to cool it naturally, and using a sterile loop or swab of the specimen, apply the inoculum to a small area of the plate, and this area of the inoculum and the plate is called the well, and from this well the different specimens or the specimen is inoculated. Then again sterilize the loop with the flame, and when it cool, or using a second sterile loop, spread the inoculum in multiple directions. This spreading of the sample will ensure a single colony of the a single colony growth. This is the example of spreading of the inoculum, that first this is the well, then this is the primary inoculation, the secondary, the tertiary and the quaternary inoculation. So this is the well. From this well the first 3 or 4 lines are drawn and inoculated, and all these lines touch each other. Then the second inoculation, which is having 3 or more 4 lines and all touches the lines of the primary inoculation. Then the tertiary inoculation also have contact with the secondary inoculation, and then the quaternary inoculation, they have slight contact with the quaternary the tertiary inoculation, so that it gives us a single colony. And this method is used if a sample is inoculated on a whole plate. This is also a simple method, and if a sample is usually sterile and have suspected of low number of microbes, so this method is used, and this is one of the simpler methods which gives us a simple identified and isolated colony. This is another method which gives us the separation of 2 samples on one petri plate, and this is the identification and the separation of 3 inoculations, 3 samples on one petri plate. So these are the different ways of the inoculation, that how sample can be inoculated on the petri plates. The inoculation of the slopes. How to inoculate the slope media, for example the Dorset egg medium or the Loeffler medium. These medium are inoculated on a slope within a bottle. So using a sterile straight wire to streak the inoculum down the center of the slope and then spread the inoculum in a zigzag pattern. And to inoculate slope in burnt medium, for example in Kliegler iron agar or the triple sugar iron, using a sterile straight wire, first stab into the butt and then use the same wire to streak the lobe in a zigzag pattern. So these are the medium. First, this is the slope. First the line is drawn and then the zigzag pattern is made, and if there is a butt, for example this is the example of the Kliegler iron agar the triple sugar iron, first it is stabbed into the base as with the help of a straight wire, then a zigzag pattern is inoculated on the slant on the slope or the slant of the medium. So this is how the slopes are the deep base is inoculated. The third type of medium is the deep medium and the inoculation of the stab medium, and with the help of a straight wire to inoculate a stab medium for the stab through the center of the medium. So if this is the center of the medium, then stab through this, taking care to withdraw the wire along the line of the inoculum without making further stab line. So if we are stabbing the wire in this direction, so with the same angle in the reverse direction, the tube and the wire should be withdrawn in the same direction and in the same line. So this is how a stab media is inoculated. Inoculation of fluid medium. How fluid medium is inoculated? So if broths or other fluid media, they are to be inoculated with the help of a sterile wire loop, or we can use with the straight wire, or we can inoculate this medium with a Pasteur pipette, depending on whether there is a colony or the inoculum is a colony, or if it is a fluid or any other specimen. So on the basis of the type of inoculum, on the basis of the sample, on the basis of the culture specimen, the different type of instruments are used for the inoculation. Using a wire loop to subculture colonies, hold the bottle or tube at an angle and then rub the loop against the side of the container below the level of the fluid. The incubation of the different type of the medium. So incubation include both the temperature as well as the environmental atmosphere, and this is determined by the type of specimen, and the pathogens may be detected in that specimen. So there is a different temperature and environmental condition for every specimen and the pathogens they contain, and it is important in every laboratory that the processing area should contain a specific chart or table which stipulate and which shows that which medium should be placed for incubation, which type of specimen should be inoculated on which type of medium, and the most bacteria cultures, they are incubated at 37 degrees centigrade to 35 to 37 degrees centigrade. The different types of microbes on the basis of the gases it use, the aerobes which grows in air and they need oxygen for their requirement. So they are incubated at the 35 to 37 degrees centigrade in normal atmospheric conditions. The anaerobes, as these are the organisms which do not require oxygen and they cannot grow in the presence of oxygen. So they require the atmosphere which is anaerobic, and this atmosphere is prepared by using anaerobic jars, the bags, or the anaerobic chamber to give such environment to the anaerobes. The third type of microbes, which are called the capnophiles, these are the microbes which require the increased concentration of carbon dioxide, but the concentration of oxygen is normal, and this carbon dioxide or excess carbon dioxide is provided by using a candle jar or a carbon dioxide incubator or a bag having carbon dioxide. The fourth type of microbes on the basis of the gases requirement, these are the microbes which are called microaerophiles, and they grow in a reduced amount of oxygen and increased amount of carbon dioxide. This is also isolated by using jars or the bags and setting the environment according to their growth. The incubation times, it varies for every individual organism, and the most time for the cultures, they are overnight or 48 to 72 hours, and the cultures for anaerobes and broth cultures, they are held for 5 to 7 days, and if there is any special organisms and that require a specific medium and a specific conditions, then these organisms, this suspected microbe is if indicated by the healthcare worker or the practitioner, so that is very helping in the identification of the microbe. This is the table which shows us the different type of microorganisms, the specimens in which these microorganisms can be found, the media used for these, and what are the different conditions for their growth. So, the Bacillus anthracis can be found in blood, skin, sputum, and usually blood agar is prepared used for this, and they are normally incubated at the 35 to 37 degrees centigrade and kept for up to 3 days with the normal oxygen requirement. Bordetella pertussis is isolated from the nasopharyngeal swab, and media used is Reagan-Lowe agar or boarded Gengo agar. Incubation time is 37 degrees centigrade, 6 to 7 days, or normal oxygen is required. Brucella species is isolated from blood and bone marrow, and it is isolated in the blood culture systems, and usually class 3 pathogens, and the PP is required as incubated at 37 degree centigrade and can be grown for up to 7 days. The Corynebacterium diphtheria species isolated from the throat, nasopharyngeal and skin species. For throat, blood agar, Loeffler agar gland and Transdale or cystine telluride blood agar is used, and normal oxygen with the normal 37 degree centigrade is required for the growth. The Francisella tularensis is collected from isolated from the eye, the lymph nodes, skin, sputum and throat specimen, and they are inoculated on the glucose cystine agar. As this is class 3 pathogen and in highly infectious can be spread by aerosols, so they required a special PPE and a special lab. Haemophilus ducreyi is isolated from the genital or lymph node aspirate, and it is a fastidious organisms. A supplemented chocolate agar is required for this specimen, and incubated at 35 to 33 to 35 degree centigrade with having 3 to 5% carbon dioxide, high humidity, and can be kept for 5 days. H. pylori, which is isolated from the gastric specimens, and media used are scuromedium, chocolate agar, modified Thiermartin agar, and this is incubated at 35 to 57 degrees centigrade, microolefric atmosphere, slightly humid for up to 5 days, and direct smear with silver or gemstone may be a diagnostic feature. Legionella species isolated from the blood, the lung, the pericardial, and the sputum, and this is the agar, selective agar, and their incubation is 35 to 37 degrees centigrade for 7 days with the ambient air. Nocardia is isolated from the brain, sputum, subcutaneous aspirate and tissue, and BHI or the HDA or serum supplemented agar or BCYE agar is used. They can be kept at micro aerophilic conditions for up to 3 weeks. And the Streptococcus streptobacillus moniliformis is isolated from the blood, lymph node and joint, and the serum sublimated medium is used, inhibited by SPS, and citrate is used as an anticoagulant in this medium, incubated at 35 to 37 microolefric conditions and hold for up to 7 days. And Vibrio species is collected from the blood, the feces, the lesion, the tissues. Blood agar, Mekonic agar, TCBS, and alkaline peptol water is used for this type of species, and they are incubated at 35 to 37 degrees centigrade with normal oxygen for up to 3 days. Now, reporting the culture, how we can report a culture? Now, the reporting of cultures, how to report a culture and the growth that is being observed on the culture media. The technologist which is examining the culture media, so he should use his skills and judgments to find out the organisms on the basis of the characteristics of the culture media, and he should know what kind of microbes have which type of the morphological characteristics. This judgment is the basic means of arriving at a result that is accurately and clinically relevant, because the clinical information along with the culturing reports is both of these can be helpful in the diagnosis and treatment of the infectious disease, and it could help the community to improve their lifestyle and to minimize their stay in the hospital and other associated issues. So, the technologists must know about the pathogens which are residing in the various body sites, and also to perform a clinically relevant workup. So, multiple questions are asked when each specimen is examined, and these specimens include questions include: what is the specimen source? So, the specimen source can lead us to the result that if this source contain any specific normal flora, so that if there is a growth on the culture plate, that what kind of normal flora may be present in this culture, and the specimen source also helps us in finding the suspected microorganisms which are usually common in the producing infections in these sites, and does this source have normal biota or it is sterile, as I discussed earlier, that if this source having any normal flora, so before reporting this normal flora is considered and that is excluded, and if any specific or significant pathogens or growth have been found, then this growth is reported. The third question, if normal microbiota is present, then what bacteria are found and what do these colonies look like? And then if there is a growth on the culture media, then what kind of the normal normal microbiota looks like on these culture plates, and how the pathogen or the infectious agents have the growth characteristics and how they can be differentiated from the normal organisms and the normal growth. What are the most likely pathogens that may be present in these specimens? That, for example, if this specimen is from the nasopharyngeal area, then what are the normal specimens, what are the normal microbiota which are present in this, and what are the suspected pathogens which can cause infection in this area, in this site. What is the colony morphology of these pathogens, how these pathogens look like on the different types of culture media, and either they produce any color or any specific colony morphology, what is the size of the colony morphology, what is the behavior of the colony morphology. So, all these are discussed when reporting cultures. Which media is demonstrating growth and what is the purpose of the media. So, during reporting the culturing of the cultures, the media is also discussed on which the organism is grown, and the purpose of the media is also discussed, why this media is used for culturing this type of microorganism. The normally sites which are sterile, so for these sites identify and report all the isolated bacteria and report their genera. So, if possible, then identify the actual species. For example, if there is any skin infection and it is caused by a gram positive such type of colonies, especially if we consider staphylococci. So, then what kind of organisms, if this is the genera staphylococci, then either it is Staphylococcus epidermidis or Staphylococcus aureus, then the actual species should also be identified if possible. The specimens which are received from different sterile body site include the blood, the bone marrow, the cerebrospinal fluid, the pleural and peritoneal fluids, and the urine. These are the specimens which do not have any sort of microbes, and they do not have any microbial flora. The sites which have normal microbial flora and their reporting is difficult and require knowledge of the patient's clinical conditions. These clinical conditions and their information is helpful in reporting such type of sites which have normal microbial flora, and this report should also indicate that those organisms for which isolation techniques have been performed. If no pathogen is isolated from these specimen, for example, if there is a fecal specimen on a selective medium, then the report should state that no Salmonella, Shigella or cholera organism is isolated, because usually the fecal specimen is cultured for these Salmonella, Shigella or cholera, and if they are grown on a specific selective medium, and certain things should be avoided reporting. For example, if there is no pathogen which is isolated from the fecal specimen, then this should be avoided to report no pathogen isolated, or to report normal bacterial flora only. These wording and these sentences should be avoided. So that was all for the today's lecture. These are the references which can be further studied for the extensive reporting and for the extensive studying of the related topics. Thank you for watching. If you have any question, you can ask. Assalamu alaikum. Allah Hafiz.